Okay, so in this next section, we're going to review the structure of RNA, compare it to DNA, and also talk about the process of what we call transcription. So, first of all, let's take a look at RNA itself. RNA, again, we have a base unit of the nucleotide. So this is the building block that actually makes up, excuse me, the RNA. Now, it is pretty much the same as in DNA. It's got the same three parts. It's got the sugar, but there is a different sugar involved. Instead of deoxyribose, we simply use ribose sugar. There is, again, the phosphate group, and we also have the nitrogenous base. Now, the nitrogenous bases are slightly different. A lot of them are the same. We have adenine, which is the same, guanine and cytosine, which are the same. But instead of thymine, we have uracil. And uracil kind of takes the place of thymine, so when it pairs up, it's going to pair with adenine. So let's take a minute here and compare the structure of DNA with the structure of RNA. And I'm just checking your notes here real quick. So what you want to do is you basically want to make a dividing line down the center, okay, with DNA on one side and RNA on the other. Okay, so in DNA we have deoxyribose sugar. In RNA we have ribose sugar. DNA is also double-stranded. It has two sides because it creates that twisted ladder, whereas RNA is only a single strand. So the teeth of the ladder steps are kind of hanging out in outer space. DNA has the four bases of adenine, cytosine, guanine, and thymine, whereas RNA has adenine, cytosine, guanine, and uracil. So if you're looking at the bases, that's kind of the big difference. In terms of location, DNA will only ever be found in the nucleus of a cell, whereas RNA is found not just in the nucleus. Now, we also have some different types of RNA. It's not that their structure is any different, but it's how they work that is different. So the first type is messenger RNA. Then we have ribosomal RNA and transfer RNA. And if you look in your notes, we're going to go through each of those. So the first one we're going to talk about is the messenger RNA, the mRNA. Its job is to read the message from the DNA in the nucleus, and then it carries the message out to the ribosome. The message that it carries is basically three base, in other words, letter codes, that determine the amino acid sequence of the protein. So you take a grouping of three of the nitrogenous bases, like AAA or AGC, and those actually code for an amino acid. Those three base codes, those three-letter codes, are called a codon. And we'll get some practice with that process here in a little bit. The second type are the transfer RNA. And transfer RNA's job is to literally transfer the amino acid to the ribosome in the proper sequence. It carries an anticodon that will match up with the messenger RNA appropriately so that we get the right amino acid in the right sequence. So this is what allows them to sequence appropriately. And the last one is the ribosomal RNA, and this actually makes up part of the ribosome, and its job is basically to help catalyze or speed up the assembly of the amino acids into the protein. It helps to position the messenger RNA and the transfer RNA properly, and it also folds the assembling protein into a proper shape. So those are the different types of RNA. That's how the structure of it is different from DNA. So let's talk about this process of making the mRNA, because that's an important process. That process is called transcription, and it occurs in the nucleus. What happens is the DNA partially unwinds and the strands are going to separate. And this is only going to happen in the region where a particular protein is coded for. So it's not all of the DNA doing this all at once. It's just a specific segment. Now, inside of the nucleus, there are floating RNA nucleotides, and they come in and they match up with the DNA nucleotides, and they form this chain. This requires an enzyme, which is called RNA polymerase. So we're going to see if this works here. Come on. Okay, now everything's gone. Sorry, I have an animation here that is not working. Pause. Okay, so I tried going in and working it that way, and the animation site is not working very well. So we'll move on. We're going to move over here to our handouts packet and kind of take a look at this. So in transcription, RNA polymerase splits the two halves of a strand of DNA. The RNA then uses one half as a template to make a copy of the other half. RNA contains a nucleotide uracil instead of the nucleotide thymine. All right, so label the DNA and RNA, and then label the missing nucleotides marked on the diagram. So let's start by labeling the DNA and the RNA. So if we look down at the bottom here, we actually have kind of this double twisting ladder here. You can see that it goes like this, and then it goes like this. Okay, so that is our DNA. This one here is a single strand. You can see how the teeth here are kind of exposed. This is our RNA. And then what we're going to do in here is actually match up. So C pairs with G. A, remember if this was DNA, it would pair up with T, but it pairs up with U. G pairs with C. Be careful here. T does not pair up with U. A lot of students get mixed up. They get on kind of U happy, but T still just pairs up with the A of adenine. All right. So, question at the bottom. In RNA, what nucleotide is always paired up with uracil? That is adenine. Now, let's also go to the next one and kind of compare our DNA replication and transcription, because these are two processes that are really easy to get confused. So DNA replication is the process by which a cell copies its DNA. During replication, both strands of the double helix are used as templates to make complementary or matching strands of DNA. DNA transcription is the process by which a single strand of DNA is used as a template to generate a strand of RNA. So we need to fill in the missing information. One row has been completed for you. So the template DNA, this first column, is what we're using to create the other two, always. Okay? Now sometimes we have it, but a lot of times here we don't. So let's look at the second row. So the second row, we know that the complementary strand is GGCC, or GGCGG. So let's go back and create the original template. Well, G pairs with C, second G with C, C pairs with G, and then two G's, we get C and C. This is important because then we need to create the messenger RNA. And in this case, the messenger RNA is going to look very similar to the complementary DNA because C pairs with G, second C pairs with G, G pairs with C, C pairs with G, and C pairs with G. So in this case, they look the same, but if you look up at the top here, they're a little bit different, and that's because we've taken the T and replaced it with the U. All right, in the third row, we've got the messenger RNA strand, so we can actually go back and actually create the template DNA. So if I have A here at the first, I'm going to have a T. Second one is a C, so I make a G. G is the third with a C. U, remember, pairs up with A. A pairs up with T. G pairs up with C, and C pairs up with G. Now we can create the complementary DNA strand. T pairs up with A, C, G, T, A, G, C. So again, these two here are the same except for that. So hopefully you're seeing that. Next one, we have the template. So let's create the complementary. That would be T, C, T, G, A, G. And then let's create the messenger DNA, which would be U, C, U, G, A, G. All right, the next row, we've got the complementary, so let's create the template. So I would have, C, T, A, T, T, C, T. And my messenger RNA is going to be G, A, U, A, G, A. Again, the only difference between these two is the T versus the U. And the last one, I have the messenger RNA strand, so let's create the template. G, A, C, C, G, A, T, G. Now the complementary strand would be C, T, G, G, C, T, A, C. Okay. Now for the question at the bottom, it says, give another example of a template DNA code that is at least 4 base pairs long, then give its matching complementary DNA and mRNA codes. What I want you to do is I actually want you to try this on your own, and I want you to have it checked by me. I want to make sure that you understand this process very thoroughly before we move on to translation.